So when you talk about dark reaction is followed by light reaction. Light reaction can be cyclic, non-cyclic. If you talk about non-cyclic in specific, Z pathway I'm talking about in cyanobacteria, it should be followed only by C3 cycle, no C4 cycle there. When you come this side, when you talk about cyclic is found in rhodospirillum. Rhodospirillum example is cyclic photophosphorylation followed by C4 cycle. Now one more is left out. Can you tell me which is left out in between these two? One more is left out now. Please help. Try to
C4.
Rhodospirillum is over. One more. Chlorobium
C4.
and chromatium. Correct? Chlorobium and chromatium. Those two will undergo non-cyclic. No. Make a note. Chlorobium as an example and chromatium as an example. These two organisms will undergo which one? Non-cyclic, where they use H2S. They use non-cyclic mechanism. Correct? But they will follow C4 cycle only. This is what is important for us to know as a difference. All are clear in this? Chlorobium, chromatium undergo non-cyclic, but C4 cycle. Whereas cyanobacteria is also non-cyclic, but it shows C3 cycle for dark reaction. That means, in brief, to tell you, cyanobacteria will show Z pathway and C3 cycle. Rhodospirillum, chromatium and chlorobium, all three will show C4 cycle only. But in that, the difference is chlorobium and chromatium will show non-cyclic, whereas rhodospirillum will show cyclic. Okay, so please have this in mind. There's a difference between the four organisms, but in three patterns. One is rhodospirillum, second is chlorobium and chromatium, and third is cyanobacteria. Okay, so if they ask about Z pathway, right? They're talking about non-cyclic Z pathway followed by C3 cycle. Usually they will not ask both the mechanisms in the same. They'll mention it very specifically whether you should answer Z pathway or whether you should write about cyclic photophosphorylation like that. So you don't need to confuse, but as a whole you should understand which organism undergoes which type of dark reaction. Okay, are you clear about this? Do you understand the differences between the three? In the syllabus also I gave you in the beginning. Sir. Okay, yeah. So now we will go for Calvin cycle. Don't worry, don't be panic. Just understand the difference. If you didn't follow, you can ask me, no problem. But be clear about this. Cyanobacteria will show non-cyclic Z pathway with C3 cycle. Rhodospirillum will show cyclic with C4 cycle. Chlorobium and chromatium will show non-cyclic with C4 cycle. Non-cyclic means there are two non-cyclics. Don't write Z pathway here. This is not, this is H2S. Today I taught you in the class now, that one. Clear? I'll give you time to go back to see. Yes, yes, ma, Sri Gauri, tell me. Today Sri Gauri is highlighted. Understood, Sri Gauri?
Yes, sir.
Okay. I want somebody to answer, no? Huh? Some Prithvika will be waiting for Sri Gauri will wait for Ritika. No, someone should answer. This is clear. I'll go ahead now for Kelvin cycle. You want a photo of this? Please tell me. I can send you.
Yes, sir. We need the photo.
Okay. Definitely. I'll send you. Rhodospirillum, cyclic, C4. Chlorobium, chromatium, non-cyclic, brackets are writing H2S. Remember, H2S is a clue. And this side, cyanobacteria, Z pathway with C3. I'll send a photo. This will help in all batches also. 1 second. I'll send it. So that you will be clear about this concept. 1 second. Let me make it clear. Maybe it's like quiz, I don't know. Let me check it. If you can follow it, it's good. No time to edit, I'm sending it directly. Okay. Now, which one I'm going to teach you? Tell me. Huh?
Kelvin cycle.
Kelvin.
Kelvin cycle.
Excuse me, sir.
Yeah. Tell me.
Sir, can we call it as chlorobium and chromatium? Can we call it as anoxygenic rather than non-cyclic, sir?
Definitely, you cannot call it oxygen. You should call it anoxygenic only.
Yeah, anoxygenic. Only we mention, is that enough?
Yes, yes.
Okay, sir.
Yes, yes. Anoxygenic photosynthesis only they will undergo. Okay. In the question
Yes,
papers,
sir.
old
Yes, sir.
members they have asked anoxygenic photosynthesis. Just take simple example. Don't write too many complications. If they ask anoxygenic photosynthesis, pick up rhodospirillum. Easy to write. You can write about a cyclic and you can write C4 cycle both together. Clear?
Yes, sir. Thank you.
Yes, yes, yes. Okay, children, I'm going to teach you C3 cycle now. Concentrate properly. What is C3 cycle called as? Calvin's cycle. C3 cycle is called Calvin's cycle. Written on the board there. Okay. Now, it starts from ribulose 5-phosphate. It starts from ribulose 5-phosphate. Ribulose 5-phosphate is 5-carbon compound. Right? Ribulose 5-phosphate comes from where? It comes from which pathway of glycolysis? See the board. HMP pathway, no? In HMP pathway, you remember you have a compound called ribulose 5-phosphate. This ribulose 5-phosphate will convert into ribulose 1,5-diphosphate. Ribulose 1,5-diphosphate. What's the meaning of this? 1,5-diphosphate means what? One phosphate is added, no?
Added.
Added. Good. So ATP is a donor. I write it like this. ATP becomes ADP. That means ATP donated the phosphate. Therefore, it became ribulose 1,5-diphosphate. The enzyme for this is called phosphorylase. Phosphorylase. Ribulose 1,5-diphosphate now converts into another intermediate compound. The intermediate compound is called as 2-carboxy. What's it called? 2-carboxy 3-keto. 2-carboxy 3-keto aragonitol diphosphate. 2-carboxy 3-keto aragonitol diphosphate. Okay. So this is an intermediate compound. This is an intermediate compound. It's an intermediate compound formed. This unstable compound. Remember, here this is a 5-carbon compound. Which one? Ribulose 1,5-diphosphate is a 5-carbon compound. Whereas this is a 6-carbon compound. Now tell me what is added here. You should know. What should be added there? 5-carbon became 6-carbon. What should be added here? Think. Many places you have studied.
Carbon dioxide.
Very good. Carbon dioxide. Carbon dioxide should be added. Can we call it as carbon dioxide fixation? Carbon dioxide fixation. We'll call it as. Okay. The role of this is carboxylation. Carbon dioxide fixation. Here the enzyme is called rubisco. RUBISCO. Short form can be asked. RUBISCO. Rubisco. Rubisco means ribulose 1,5-diphosphate carboxylase. I'm writing short carboxylase here to remember. Carboxylase. Okay, rubisco means ribulose 1,5-diphosphate. Me means diphosphate means biphosphate also you can call it as. That's why it's called rubisco. Di means Bi, both are same. Di, bi, both are same. Rubisco, ribulose 1,5-diphosphate carboxylase will fix the carbon dioxide, and thus we got a 6-carbon compound called 2-carboxy 3-keto alabinitol biphosphate or diphosphate, both are same. This unstable compound will form 2 molecules of, see the 6-carbon, remember children, it's going to form 2 molecules, 2 not 1, 2 molecules of 3-phosphoglyceric acid, 3-PGA, I'm writing short form, 3-phosphoglyceric acid, 2 molecules. How many carbon 3-PGA will be, tell me?
3 carbon.
3 carbon, very good. So 2 molecules means did not satisfy 6 carbon totally? Yes. 3 carbon this is, no. Just write 3 carbon here. You will understand. 3 carbon. 3 carbon into 2 means 6 carbon. It's got split. This compound got split into 2 molecules of this. Are you clear? Yes?
Yes, sir.
Yes, sir.
Okay, okay. This now will convert into 2 molecules of 1,3-diphosphoglyceric acid. 1,3-diphosphoglyceric acid. Little of glycolysis comes here. What can you guess what should be added here? 3-PGA become 1,3-dipga. Yes, tell me.
Phosphate.
What is it?
Phosphate.
Phosphate. Good, very good. So we must add ATP here. ATP becomes ADP. Phosphate is added. So we got, means I'll write it in yellow color so we remember. ATP becomes ADP. Can anyone tell me where we got this ATP from? This ATP we got from where? Can you tell me? Guess very fast.
Light.
From the light reaction. Very good. We studied no light reaction. There we got ATP. No, that ATP is accounted here. It's used here. Think like that. Okay. The name of this enzyme is called phosphoglycerokinase. Phosphoglycerokinase. We got 2 molecules of 1,3-diphosphoglyceric acid. Finally, this will become 2 molecules of, converts like this. It becomes 2 molecules of glyceraldehyde 3-phosphate. Glyceraldehyde, remember, glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. 2 molecules of glyceraldehyde means what? 1,3-dipeptide becomes glyceraldehyde. Means, I'm sorry, I have to show the. Please remember here, hydrogen is added. That means NADPH2 is used. NADPH2 is used. Hydrogen is used. Reduction became NADP. This is clearly a step called reduction. This previous step is called phosphorylation. And here it is called carbonized fixation. What do we call this as? Carboxylation. Now can you tell me what are the 3 important steps you learned? Overall, carboxylation, phosphorylation and reduction. These are the important steps in Calvin cycle. Though you have so many steps, but we highlight, we give importance for Calvin cycle in these ways. What are the different things? Carboxylation, phosphorylation, reduction. Any doubts, children, till here? All have followed till here. The enzyme for this is glyceraldehyde 3-phosphate dehydrogenase. Yes. Did you follow till here? Want me to
Yes,
repeat
sir.
this part? Yes. Or you followed? Vansha. You don't answer.
Yes,
I
sir.
understood. You followed.
Yes, sir. Understood.
Okay. Fine. So just keep observing. It's a revision once again for you. This process, whatever we learned from here, no, this process repeats 3 times. So I'm repeating once again, just observe it. Means we should write like this. See the diagram? We should write like this. From here, this whole thing is repeated 3 times. So we call it as C3 cycle. What is repeated 3 times? Ribulose 5-phosphate, 3 molecules. See the board. 3 molecules of ribulose 5-phosphate converts to 3 molecules of ribulose 1,5-diphosphate. Right, 3 everywhere. 3 ATP molecules are used. 3 molecules of carbon dioxide is fixed to give you how many? 3 molecules of what? Somebody tell me this name. 3 molecules of what?
2-carboxy 3-keto arabinitol diphosphate.
2-carboxy 3-keto arabinitol diphosphate intermediate. So here also I should say, if one gives 2, 3 molecules will give how many? That means 3 2s are
6.
6. Yes, children, good. So everywhere I'm writing 3 3. Can you guess where we came across such a concept? Somewhere like this we learned now, where we should write additionally like this in square bracket afterwards.
EMP
pathway, the very first pathway. Very good. Okay, I'm happy you remember this. Therefore, what should I write here, children? 6 molecules of glyceraldehyde 3-phosphate is formed ultimately. You agree with me? See the board. See the board here. 6 molecules of glyceraldehyde 3-phosphate is formed. Among the 6 molecules, okay, see the board, very important here. Among the 6 molecules, 5 molecules will go for regeneration. What will regenerate? What will regenerate? What is this?
Ribulose
Plus
5-phosphate.
5-phosphate. That means only 1 molecule is left out, my children. 1 molecule is left out. This 1 molecule only will help in forming the glucose. This 1 molecule will help in what? Forming the glucose. I do not place a right in this part of the board. You should understand here. I'm writing it this side. What I'm writing here, see. I'm using this part of the board now. What I want to explain here, 1 molecule of glyceraldehyde 3-phosphate. Understand slowly. 1 molecule of glyceraldehyde 3-phosphate combines with dash. You should tell me this one. To give fructose 1,6-diphosphate. You just told me, just sometime you told me EMP pathway, no? Recollect and tell me. Glyceraldehyde 3-phosphate plus dash will give you fructose 1,6-diphosphate. Tell me. Think. Reverse of glycolysis. I've given the clue. Reverse of glycolysis. You reverse it. Fructose 1,6-diphosphate will give something. No, I'm reversing it here. Those two will give fructose 1,6-diphosphate. Which is the other one? Think. You know the answer. Can anyone tell me EMP? Start with EMP. I'll help you. Glucose becomes. Tell me.
Glucose 6-phosphate.
Yes.
Then fructose 6-phosphate. Then
Yes.
fructose 1,6. And then splitting cell, glyceraldehyde 3-phosphate and DHAP.
Ah, you got the answer there. What is it?
Splitting, aldolase enzyme.
Huh?
Splitting.
No, ma. Glyceraldehyde 3-phosphate. DHAP.
DHAP.
Yes, that's the answer here. DHAP. DHAP is actually what is reverse. This will split into these two now, but here these two will combine and give this. All are clear in this? Are you all clear in this? Glyceride 3-phosphate plus DHAP gives you fructose 1,6-diphosphate. Reverse glycolysis now. Who told me this answer? Reverse it again now. Go back now.
Glyceride 3-phosphate plus DHAP will give you fructose 1,6-diphosphate.
Okay.
And then fructose 6-phosphate, then glucose 6-phosphate, and then glucose.
Understood. That's all. Go from here now. Reverse it. Fructose 6-phosphate, glucose 6-phosphate. Then write glucose 1-phosphate, children. Glucose 6-phosphate becomes glucose 1-phosphate. Glucose 1-phosphate will become later on starch or glycogen, whatever. See, I'll repeat once again. Glyceride 3-phosphate plus dihydroxysterone phosphate gives fructose 1,6-diphosphate, followed by fructose 6-phosphate, then glucose 6-phosphate. But one thing you should remember, glucose 1-phosphate. Glucose 1-phosphate. That will complex to starch or glycogen. Thus we get food synthesis. Any doubts in this? All of you understood this till here?
Sir, when they ask to write the cycle in this manner only, we have to write, sir.
This way you should write, yes. 5 molecules
Like other
regenerating ribulose 5.
no, the order, sir.
Huh?
Proper order, or we should write in cyclic manner only, or like this only we can write, sir.
Cyclic manner only you should write, ma. Better. Whatever written on the board, no, like this only you write. Use the full sheet. Okay.
Okay, sir.
Okay. That is better. If you write it in one below the other, you can write it. Doesn't matter. But see how you are comfortable. No problem.
Okay, sir.
Okay. All of you follow till here. Yes.
Yes, sir.
Okay. So what are we left out with in photosynthesis then? Only C4 cycle. 2 o'clock, please attend the class. We'll complete C4 cycle, and I have to plan for bioenergetics also partly. Okay. Okay. Have your lunch and come at 2 o'clock. Or you want me to prepone the class? Tell me. 2 o'clock is fine. Sampriti, please check attendance, ma. We have only 1
Yes,
minute.
sir.
Okay.
Take it, sir.
Thank you. Thank you. All of you followed. I'll end the class. Only 1 minute is there. Anyway, we'll end the class. Any doubts are there? You can ask me now. Then we'll continue at 2 o'clock. We'll complete with the photosynthesis. We'll finish in half an hour. That is left out. Then we bioenergetics. I'll start with how much energy you have in C. Okay, now.
Yes, sir.
Afterwards, I'll give you some questions also in further classes. You can study for that. Okay. Shall we stop now?
Yes, sir.
Okay. Okay. See you then. At 2 o'clock, please attend the class. By prepone, I'll inform Yashas. Yashas? Yashas? Is there? 1:30 is okay for you people?
Yes, sir. Yes, sir. Yes, sir. You can
Okay,
take it.
anyway, I'll inform. I don't know. I have to go back to department and see. I may prepone if at 1:30. Okay. Fine. Fine then. See you. I'll send a photo of this one. I'm sending it.